Oke, sudah bisa. Saya overhost ya.
Oke. Push. Okay. Wait, now I will push. Pak Adit ya, Adit. Belum join ya Pak Adit ya? Belum kelihatan nih. Awang, Hideo Kato, Usa, mana Usa ya? Usa. Hello. Good morning.
Good morning.
Good morning, everyone. Good morning, Prof. Usa. How are you?
Good. How are you? Okay, let me change the background first.
Okay.
Good morning.
Good morning, Prof. Hideo Kato. How are you?
Thank you. I'm fine. Thank you. How are you?
I'm fine. I better wear mask. So thank you for spending your time to deliver this webinar, Prof. Usa and Prof. Hideo Kato. Today, the number of participants who, excuse me, who register are 900. Excuse me.
Wow.
And hopefully many will join. And we also do the live stream YouTube now. So because of our Zoom is only limited for 300, then others will join via YouTube. Okay. So I think as a preparation, would you mind to Prof. Hideo Kato to try the share screen? And then, hello, Prof. Usa.
Hi, Professor Adi and Echo.
Finally, we
Nice
meet.
to meet you.
Very nice to meet you. Wait, I just take a drink. Prof. Hideo Kato, you can try your share screen, and afterward, Prof. Usa. I just take my drink first.
สวัสดีครับอาจารย์
สวัสดีค่ะ Very good to see you, Adi.
อาทิ taking a drink อาจารย์
Can you see my
You
screen?
haven't changed at all.
yeah we can see your screen Hideo thank you
Okay.
nice can you just stop it and Professor Usa will try to
Okay.
yeah thank you
Okay.
yes อาจารย์ we can see your PowerPoint thank you อาจารย์
Thank you. Oh, looks great. I mean the share screen. Now I will invite others to join, I think. Wait.
Actually, I would like to update Ajahn Usa and Associate Professor Hideo. The number of participants is almost 1,000. Like to be exact, is 974 participant register. Yeah.
Wow,
Okay.
so many.
Wow, congratulations.
Oh, thank you, Ajahn, for your support. It won't be able to happening without your support and Hideo's support. So thank you.
Yeah, thank you for both
Oh,
Prof.
it's
Hideo and
my
Prof.
pleasure.
Usa. No, I think as Pak Adit is here or not, Pak?
I will ask him. Wait a moment.
Okay.
Oh. Cool. Oh.
So each speaker will talk about 20 minutes, and then it will be moderated by our colleague from Faculty of Pharmacy, Mr. Adit. And then afterward, there will be question and answer session, and will be moderated by him.
Okay.
Okay.
Pak Adi,
Yeah.
would you mind to allow Bu Farida to join and also Adit to join the Zoom? Because it's
Okay.
like
Okay.
waiting. Yeah.
Okay. Adit ya. What's what's his name, Pak? Pak Adit kok nggak ada ya? Farida juga nggak ada. What's his or her name here?
Farida is already in this room, Pak Adi.
Kamera, Bu.
Oh, oke. Okay. Halo, Bu.
Oke.
Pak Adit, what is...
Morning.
Morning, Bu.
Oh, enggak denger ya, Ming ya?
Adit is not here yet, I think, Pak. Andrew Raditya. Muhammad Andrew. I don't see his name.
Oh, belum Pak.
Oh,
Bentar, Pak.
that's okay. Tadi nggak ada. Okay. Then Mr. Vera Kit is already here. I will admit. Then okay. So only Miss Dr. Muhammad Hafiz Abdul Aziz is not here yet. Assistant Professor Firakit is already here. Good morning, Assistant Professor Firakit. How are you?
Hello, good morning. How are you?
Good morning.
Hi.
Hi, I'm Adi as the chairperson of this global webinar. Thank you for Assistant Professor Firakit to support this webinar. Today we have 978 participants registered, Prof. So hopefully this webinar will be successful. I think as the preparation, would you mind to try share screen, Prof. Rakit?
What's that?
Try to share screen. Share screen.
Okay.
Okay.
So just trying.
Yeah.
Okay.
Make sure that it works. Yeah.
Okay.
Just make sure that it works. Okay. It looks great.
Okay. Good.
Okay. Good morning, Pak Adit. Ready, Pak?
Good morning.
What time do you want to start, Pak?
Yeah, we can start actually.
We can start now. I'll give the host.
We can
Pak.
start.
Wait. I don't forget to pin the speakers, ya, Pak. Later, one by one. I think.
I will get the.
I will give you a host.
Admin.
Yeah. I
will be the host. Okay.
Yeah. Wait. Okay, you are now host. So.
Sorry.
Yes,
Sorry.
Prof. Video. Yeah.
So I can change background. So I don't know how to
go to
change.
Zoom. Go to Zoom and then settings.
Yeah, because I tried this one, but reverse.
Okay. If not, now it works. It works now. Oh, yeah. It was. Yes. Now it works. Now you have the virtual background.
It is okay.
Yeah, it is. Now it appears that you have virtual background.
Okay.
Okay, Pak Adit, we will start, ya, Pak. So the brief rundown is I will open to introduce speakers and also the moderator, and then I will give time for the Dean, Bu Farida Suhud, to deliver a speech, and then afterward would be group photo. And then afterward, I will give the fully time to you, ya, Pak Adit, ya. Okay?
Okay, Pak Adi. Okay.
Okay, now I will admit all. Oh, you should admit all because I'm not co-host now. Or Sandra, can you admit all?
Yep, I will enable.
Okay. Okay, now let's do it. Wish all the best for us. Yeah.
Ada Ubaya.
Okay, I think we should. Okay, let us start now. Good morning, everyone. How are you? I hope all of
Morning,
you
good.
are in healthy condition as we are now. We are very excited today because we will have 4 speakers from over the world. And we will also have a very interesting topic about pharmacists' contribution in the healthcare system beyond advance to bed. Today we will have Professor Hideo Kato from Mie University Graduate School of Medicine, Japan. How are you, Prof. Hideo Kato?
I'm good, thank you.
Also, we will have Assistant Professor Ferakit Bun Kanokbong, PhD from Chulalongkorn University, Thailand. Good morning. How are you,
Hello,
Assistant
good morning.
Professor? Good morning.
Good morning, I'm doing good, thanks.
Also, we will have Associate Professor Dr. Usa Taiklet Kau from Faculty of Pharmacy, Mahidol University, Bangkok, Thailand. Good morning, Sawadika.
Good morning, everyone. Nice to see you all. It's my pleasure to join the conference.
Thank you, Prof. Usa. Also, we will have Muhammad Hafiz Abdul Aziz, PhD from the University of Queensland, Brisbane, Australia. So we have from Japan, we have from Thailand, and we have from Australia. Not to forget, I also would like to welcome all participants who are available, even though now is weekends, but you are now here to join our webinar. And then I also would like to welcome and thank you for the support from the Dean, Dr. Farida Suhud. Thank you, Bu, for joining. Thank you
You're
for supporting.
welcome, Pak Adi.
Also, I would like to thank you, Pak Aditya Trias Pradana, as a moderator. He is now currently the PhD candidate from the Faculty of Pharmacy of Ubaya. Last but not least, I also would like to thank you, Pak Eko Mingming, as his interaction and communication with me enable this webinar is possible to be conducted. Thank you, Pak.
Thank you, Pak Adi.
Okay, now as a first session, I will give time for Dr. Farida Suhud to deliver her opening speech. Please, Bu.
Thank you, Pak Adit. Pak Adit sampai Pak Adit. Good morning, ladies and gentlemen, speakers, and good morning to our audience joining us online from all over the world. First, may I thank each of our speakers for taking part in this event. Associate Professor Hideo Kato, PhD from Mie University, Japan. Arigato gozaimasu. Assistant Professor Wirakyat Bun Kanok Wang, PhD, RPH from Chulalongkorn University, Thailand. Muhammad Hafiz Abdul Aziz, PhD from the University of Queensland, Australia. Associate Professor Dr. Usa Chailekiao from Mahidol University, Thailand. The quality and caliber of all speakers this year is outstanding. I also want to thank all of you for connecting this morning, and I hope you are looking forward to the discussion as much as I am. As per this morning, there were 978 participants who will be joining us in this symposium. The theme of our international symposium is Pharmacist Contribution in the Healthcare System, Beyond a Bench to Bed. We should be clearly aware that pharmacy is a broad discipline covering the life cycle of drugs from the point at which the targets are discovered to when they turn into medicine, encompasses their delivery, distribution, dispensing, and management. This means that the discipline of pharmacy cuts through all aspects of life sciences, from drug discovery and development to clinical sciences and healthcare services. Our symposium will cover an important issues during the life cycle of drugs. In accordance with the vision of Ubaya, that is the first university in heart and minds, and also the vision of Faculty of Pharmacy, Ubaya, that is to be the pioneer for transformation of pharmaceutical sciences and pharmacy practice education to improve society's health and quality of life. This international symposium, one among many efforts to make these visions and mission come true. This international symposium is held to celebrate the 55th anniversary of Ubaya. It is part of our concern on contribution of science and technology for enhancing nation productivity and competitiveness in Indonesia. Hopefully, we all can significantly give more contribution to the nation advancement in the not too distant future. To all of our distinguished guests and participants, thank you for being here. Welcome and enjoy the conference. Ladies and gentlemen, let me once again thank our fantastic speakers for giving their time and insight today. And I hope all of you enjoy the conference. Thank you.
Thank you, Dr. Farida, for your inspiring opening speech.
You're welcome.
I think now we can start our webinar as Dr. Farida has opened it. Now I give a time for the moderator, Mr. Aditya, to start and direct the webinar. Pak Aditya, time is yours, Pak.
Okay, thank you, Pak Adi. Yeah, good
Wait.
morning.
I think because, before we start, I think something important is group photo.
Oh, okay.
Almost forget.
Okay.
Because one of the speakers has a limited time, so we do the group photo now. Sandra, would you mind to comment, please?
Okay, I will take a group photo. Would you mind to turn on your camera, everyone? I will wait until the count of 5. 1, 2, 3, 4, 5. Okay, I will take a photo from the first page first. 1, 2, 3. Thank you. And then moving to the second one. 1, 2, 3. Thank you so much for turning on your camera, guys. And third one. think that's enough. I think. Thank you.
Thank you, Miss Sandra, for taking photo or screenshot for us. Okay, now we come to the main session. As I said, I will give the time for the moderator, Mr. Aditya Trias Pradana, PhD candidate, to start and direct this webinar. Please, Pak, time is yours.
Okay, thank you, Pak Adi. Good morning, ladies and gentlemen. Glad to meet you today in Ubaya Global Webinar Series 2023. This is celebrating the 55th anniversary of University of Surabaya. So the topics of our webinar today is pharmacist contributions in the healthcare system beyond bench to bed. First of all, I would like to welcoming our four speaker today and thank you for your calm and willingness to share knowledge with us today. So I will explain the timeline first. So each of presenter will present the topic for 20 minutes today. And after the second and fourth speaker, we will open the Q&A session. So audience can ask the question directly by raising the hand or write in the chat box. So the presenter will be answer the question after I read from the chat box. Maybe like that. And we will continue. We will start the first presentation today. So I will introduce our first speaker. Wait a moment. Let me pin first. Okay. Okay, so I will introduce Associate Professor Hideo Kato, PhD. So he was born in 1988 and he got his PhD in Doctorate of Medical Sciences in Aichi Medical University, Aichi, Japan. So Dr. Hideo Kato has so many research, especially in the area of relationship between pharmacokinetics and pharmacodynamics PKPD of antimicrobials. And he also has some experience in the infection cases as a clinical pharmacist right now. Because Dr. Hideo Kato also right now active as a researcher in the Department of Clinical Infectious Disease, Aichi Medical University School of Medicine, Aichi, Japan. Is it correct, Dr. Hideo? And also, he's active as an associate professor in the Department of Pharmacy, Mie University Hospital, Mie, Japan. And then he also as an associate professor of Department of Clinical Pharmaceutics, Division of Clinical Medical Sciences, Mie University Graduate School of Medicine, Mie, Japan. He has so many publications, around 100 publication, and most of them about animal behavior, antimicrobials, and pharmacokinetics. Then his newest publication of Dr. Hideo Kato are about effectivity of targeted ampicillin and cefazolin in also pneumonia therapy, safety evaluation of COVID-19 therapy, and gentamicin pharmacokinetics in the infant patient. So I believe that he will be the best person to talk about the first topic today, animal studies and drug dose optimization concept and implementation. So Associate Professor Hideo Kato, PhD, please, the time is yours for the first presentation.
A very good morning, everyone. Thank you for the introduction, and also thank you for inviting me to speak. And thank you all in the audience for your attendance. So, share my slide. What? Where? Sorry, just one, please. Yeah. Can you see my slide?
Yes, We can see it clearly.
Okay. Oh, thank you. So I'm Hideo Kato, the Associate Professor in Department of Pharmacy, Mie University Hospital. I have conducted animal study of pharmacokinetics, pharmacodynamics, efficacy, and toxicity using rodent models, including models for antibiotic efficacy analysis. And I'm going to talk about mouse models commonly used to study the activity of antibiotics and animal studies we have conducted. So let's advance the slides. The objective today is that I'm going to be covering how we establish infection mouse model, and then how do we use them to characterize antibacterial compounds that are used or will be used to treat antimicrobial resistant infection. And so I'm going to be focusing on lung and thigh infection models with neutropenic mice. These types of models are used to evaluate world model to generate models with World Health Organization priority, gram-negative, and positive pathogens. And they are commonly used for antimicrobial drug discovery testing. We have a number of models and that are not included. I will not talk about today, including urinary tract, wound dermal, and vaginal gonorrhea models, but they are conceptually similar. And so I think this will give you a good insight on how the models are developed and how they are used for efficacy. So let's go to the next slide. Before starting out talking about animal studies, I point out non-clinical infection model, and that's and that can be performed with widely used, and the models include static time chem models, chemostat, hollow fiber model, and animal studies. Each model has some studies objective. Although I do not talk about hollow fiber model in detail today, this model covers all study objective and are currently used by most pharmaceutical companies to evaluate the efficacy and pharmacokinetics of antibiotics. However, this model does not have immune system, so that there may be some gap regarding the efficacy between this model and human. Laboratory animal models have been used for decades to identify effective dosing regimen for clinical trials. Also, dosage, drug clearance, including metabolism, and other factors often differ considerably between animal and humans. In vivo models play a critical role in characterizing the PK/PD for antibacterial agents. Animal models provide an in vivo infection environment and anatomical barriers that are difficult to reproduce in vitro. Animal infection models can forecast drug efficacy in patients, and the probability of regulatory approval increases with the probability of PK/PD target attainment. I'm focusing on mice, but there is, of course, other animal species that can be used to evaluate an antimicrobial agent. And the reason I'm talking about mice is that they are most commonly used for early testing because they are reliable and relevant mammalian species that is commonly used for testing PK/PD, efficacy, and toxicity. There is established models that are widely used across the industry, and there is published data available for most approved drugs using these models, in particular the thigh and lung models. Most importantly, the exposure-response relationship established in mouse, thigh and lung models translates to efficacy in humans. So these are really important models throughout the discovery and development of antibiotics. I'm going to talk about the thigh and lung model, and these are conducted with neutropenic mice. They are rendered neutropenic with cyclophosphamide. The neutropenia eliminates the activity of the host human system so that the pathogen can grow quite well. And the antimicrobial effects that is observed in the models are due to the test articles activity. The cyclophosphamide is administered at 150 milligram per kilogram, therefore prior to infection, and then again at 100 milligram kilogram, they want prior to infection. Mice are infected on day 0, either by intramuscular or by intranasal injection, and then test articles and doses initiates starting at 2 hours after infection most commonly. At other tests, the dose time depending on the test article, the half-life, etc. There are 2 animal types of animal groups. There is one control group, and that is sacrificed at 2 hours after infection for the baseline count. And you are going to hear about the baseline counts a lot. And then for the endpoint, to look at the treatment effect, animals sacrificed at 26 hours after infection. The tissues are harvested and the bacteria are enumerated with the dilution most. The animals most typically are female. Again, they are neutropenic, and most commonly I see our mice 6 to 8 weeks old. We use 6-week-old mice. This is an example of how we established the infection model, and this was conducted with MRSA and Streptococcus pneumoniae. MRSA has 3 mRNA of daptomycin. The first step to generate a reproducible model is to determine the inoculum concentration. We have 2 time points, 2 hours corresponding to that 2-hour baseline count, and then 26 hours. And we measure the bacteria counts with dilution plating. We did this for each bacteria. What we are looking for is a baseline counts of approximately 10 to the 6 to 10 to the 7 CFU per mouse in the thigh and lung models, and about at least 1 log or 2 log increase in count, preferably 2 log for 10 to the 8 CFU per lung at the 26-hour time point. So in both infection models, you can see the establishment of infection. And the second step in establishing the model is to evaluate a standard of antibiotic to use as a control, and then also assure that the model is dose responsive effect. This shows the evaluation of meropenem in thigh model infected by Klebsiella pneumoniae, and the type of data that we can recover from these models. On the vertical axis is a change of bacterial counts obtained in the treated mice after 24 hours compared with the numbers in the study control animals. And on the horizontal axis is dose group going from low to high. A bacterial count in the control group increases 1.4 logs, and we see a very nice dose response of reduction in bacterial counts by meropenem in the thigh model. One of the points that I wanted to make is what you consider significant effect. Significance is evaluated using ANOVA to compare to the control count, because what we are looking for is an antibiotic bacterial effect, reducing the counts relative to the start of the dose period. Moreover, we can calculate the dosage of meropenem at the stasis, 1 log reduction, and ED50 using Emax model. Consequently, the calculated regimens associated with stasis and 1 log kill dose 58 and 146 milligram per kilogram every 8 hours, respectively. The site and lung infection models are very important for lung ordering hits and for read optimization and comparing test articles for their potency. Especially as the site model is also very important for establishing exposure response correlation for predicting efficacious dose in humans. So it has a translational role and a clinical development role. I want to go through these types of PKPD evaluation to estimate target exposure values if it is based on the principle of PKPD. The antimicrobial efficacy is the outcome of the test article exposure, that is the dose in the PK and also the MIC that represents organism's susceptibility. In other words, PKPD parameter is associated with the antimicrobial efficacy. The PKPD parameters include the time above MIC, the AUC bar, or the Cmax by MIC, and these parameters correlate with the efficacy in mice and also in humans. In this graph, that is showing the concentration time profile. As expected after dosing, you have a sharp increase in the concentration, and then the concentration decreases over time. The time above MIC is the time that the concentration is above the MIC. There is a Cmax by MIC value, that is the maximum concentration of the MIC, and then there is AUC of the MIC value. Characterization of the PKPD supports the optimization of the dose regimen in humans. First study is the pharmacokinetics evaluation measuring the concentration as a function of time. In a PKPD evaluation, the pharmacokinetics are measured in the infection model because the infection can alter the pharmacokinetics. In a generally range of dose that span the step region of the dose response curve are used. This slide shows the antimicrobial pharmacokinetics profile of sulfamethoxazole at single dose of 86.6, 130, and 390 milligram per kilogram. Like shown in the table, from this data, PK parameters such as Cmax, Tmax, and AUC are estimated. You can see that with the doses of sulfamethoxazole increase, the Cmax and AUC increase. Next, with the PK and the dose ranging results established a dose fractionation where a range of dose concentration are tested at different dose frequencies as well as divided dose frequency. In this case, time model infected by Staphylococcus aureus isolates were administered sulfamethoxazole in 1, 2 over 3 equally divided dose over 24 hours. The doses selected for the dose fractionation study are based on the result of the dose range study and PK study. In all those frequent dosage regimens showed numerically higher antimicrobial activity than the single dose regimen. So it seemed likely that time above MIC was the most predictive PK/PD parameter. Finally, the efficacy and the pharmacokinetics are analyzed. The PK such as the AUC, the Cmax, and the time above MIC are estimated at all of the dose concentrations tested, and then the data are fit into curves looking at the bacterial counts as a function of the PK/PD parameter. In this case, on the upper left, the time above MIC, on the right, the AUC per MIC, and on the left below, Cmax per MIC are shown. The data are fit and the regulation is evaluated to determine the goodness of the fits. For sulfamethoxazole in Thai model infected by Staphylococcus aureus, you see that the goodness of fit using Cmax by MIC is quite poor, with R-square value of 0.53, whereas the time above MIC over AUC by MIC had a much better fit with an R-square value of 0.71 or 0.69. However, the dose response curve gave the impression to be truncated at time above MIC of 100%, and no effect at time above MIC less than 100% is admitted. Therefore, time above MIC of sulfamethoxazole could not be the best PK/PD parameter to explore the optimal dosing regimen. As a conclusion of this study, the AUC per MIC was a better predictive PK/PD parameter for sulfamethoxazole. Those correlations for killing correlate with efficacy in humans and are used to establish those targets. Since this is clearly a very important measurement, there are now guidelines for how to conduct these experiments to generate robust data. These guidelines cover the preparation of inoculum, the bacterial burden, the time for the infection period, and the numbers and types of strain to be used. Now, so to recap the key points, the Thai and lung models are workhorse models for drug discovery and aid the establishment of those for the clinical development. The PK/PD parameter, that is time above MIC, AUC per MIC, and Cmax per MIC, correlate with the efficacy in mice and humans. And there is not a step-established protocol guidelines for conducting these models to establish PK/PD targets. Finally, I introduce our laboratory to you. There are a cultivated room, pharmacokinetics laboratory, and the molecular biological laboratory in our department, and shared animal room in our university. Especially our strength is to be able to quantitate drug concentration in both animals and the human using mass spectrometry and chromatography. Therefore, we can perform PK/PD study on various drugs except for antibiotics. I want to thank you all for your attention. Well, this brings me to the end of my presentation. Thank you once again for taking the time to join today's presentation. Please do not hesitate to contact me if you have any questions. Thank you.
Thank you, Dr. Hideo, for your inspiring talk. We will open the Q&A session later after the second speaker, Dr. Hideo. So please wait a moment. Thank you very much.
Okay, thank you so much.
Okay. Okay. Good morning, Achan.
Hello. Hi. Good morning. Ajahn.
Hi. Nice to see you again.
Yep. Nice to see you.
Achan.
Hi.
Okay, so we will continue with the second presentation from Assistant Professor Virakia Punkanupong, PhD registered pharmacist. So he has finished his Master of Engineering and Doctor of Philosophy in Chemical and Biochemical Engineering, Rutgers, the State University of New Jersey, the United States. Right now, he is active as a lecturer in Department of Pharmaceutics and Industrial Pharmacy, Faculty of Pharmaceutical Sciences, Chulalongkorn University, and also as a secretary committee member in the Administrative Committee of Masters of Science in Pharmacy or Doctor of Philosophy programs. He also is an assistant manager and a pharmacist in the Community Pharmacy Laboratory at the Salah Faculty of Pharmaceutical Sciences, Chulalongkorn University. Dr. Virakia's research interests are about pharmaceutical and biopharmaceutical engineering and processing technology, particle engineering, and pharmaceutical solid dosage form manufacturing processes. And also about pharmaceutical development and quality system within quality by design or QBD concepts, and also numerical simulation of granular material using discrete element method or DEM. He has so many publications in the field of formulation and industrial pharmacy. The new publication is about production and investigation of astaxanthin self-microemulsifying tablets, including the implementation of mixture design, and also research about scaling up and behavior of materials by four-plate mixture and others. So today, Dr. Virakia will present about implementation of a quality by design approach for pharmaceuticals and other healthcare products development. So for Assistant Professor Virakia Punkanupong, PhD, registered pharmacist, please, the time is yours.
All right. Yes. Thank you, Aditya. Thank you for the introduction. So first of all, let me share my presentation first. Do you see my screen right now, right?
Yes.
Okay.
Maybe you can full screen it. Okay, thank you, Ajahn.
Okay, so first of all, I would like to thank Mr. Aditya who invited me to join and give a talk to the University of Surabaya seminar series, this webinar today. So it's my pleasure and honor to be invited to give a presentation in this seminar. So hopefully you will get some useful information from the topic that I will talk. So the topic that I would like to, let me call it, share the information with you guys is about the applying or implementation of a quality by design method for the pharmaceutical and actually for other healthcare product development also. So what is actually a quality by design approach, and what are its building blocks for the quality by design methodology? Okay, and in this talk, I will also cover the implementation or example of how to apply the quality by design approach to the pharmaceutical and healthcare product development. And if the time allows, I will give you some research example that used the QBD and design of experiment approaches to develop their commercialized products. Okay, so I would like to begin with this slide first. So usually in pharmaceutical industry or pharmaceutical manufacturing, they use a what we call a batch manufacturing procedure, right? What that is you just put an input into the process and then you get the output as a batch or per lot. And what do we do the quality control is that after we get the output, let's say, for example, you do a compression of a tablet, okay? You do a quality control by sampling, using a statistical sampling to sample just a few sample from the whole population, right? And then you test some of the quality control items. For example, you test the hardness, weight of the tablets, you measure the disintegration time, you measure the dissolution profile, something like that. And then if according to the official monograph or pharmacopoeia, right? So after you got a test result from that sample, if the test result, the quality of that test result is passed, so you assume that the whole population of the tablet batch is the same quality, the quality is passed, right? But if the results are failed, you assume that the whole lot or the whole batch are failed, right? So this is what a quality control paradigm that we call it's quality by testing, right? So there is a disadvantage of the quality by testing is that if the result is passed, you cannot like 100% assume that all of the population or every individual tablets is going to meet the quality requirements, right? And if it is failed, actually some of the samples or some part of the population might pass also. So it depends on your statistical analysis and sampling also. So they are said to be that this is the quality by testing method cannot be like fully reliable. So that in the recent era, many of the regulatory authorities encourage to use the quality by design approach instead of testing and sampling method. So what is actually the quality by design? It is a systematic method to develop. Need to your objective or your quality of the product that you are developing first. So what is your quality of the products that you want, okay? And it says this approach focuses on the product and process understanding. You need to understand how the materials or your process affect your quality of the products. Okay, and also you need to do a process control by some strategy. So everything is based on the scientific evidence and also the quality risk management. So you need to use the risk analysis and risk management to analyze or to help doing the quality by design things also. So right now, FDA or ICH guidelines say that the quality cannot be tested from the product or the process. We cannot rely on statistical sampling and test anymore. The quality should be built or must be built by design into the product and process. So that's why the quality by design approach becomes more important. So here's the slide showing about the quality by design concept. So what you already know is called the knowledge space from what you know, like prior knowledge, experience on your work, or you are reading and doing some literature reviews, right? And we also have a more smaller area, what is called the design of experiment or characterized space area, that you already screen and know the effect of the input variables to be affected to your product. Okay, so what we would like to establish or build is what is called the design space within here. So the design space is a range of independent variables that you know, okay, and you prove that within this design space range, it's gonna make your product within the quality acceptable range. Okay, but there is a more stringent or more smaller range within the design space. We call it a operating or control space or manufacturing space. So the reason why we just manufacture the product within the design space is that we can control the uncontrollable factor even better in here. So this is a concept that why should we establish the design space. So how quality by design should be started? So when you look at some pharmaceutical unit operation or any process, you can look at it as a simple process. So you have a input materials, which is a controllable variables that you can know, right? So we would call it as a critical material attributes. Some variable are not critical, some are critical. So you need to use a risk management to identify which are critical and which are not important. Okay. So for the process or each unit operation, you also have a process parameter. So again, you need to identify which process parameters are critical and which are not. Okay. Besides of the critical material attributes and the critical process parameter, there are also always has a uncontrollable variables, or you can call it noise, which is a residual that you can control and beyond of your control. So that would be give the effect to your quality of your product also. So the output product is a function of the critical material attributes and the critical process parameter. So we would call that output quality is a critical quality attribute. So you would define or predefine that your product, even injection, a tablet or capsule, they should have a pre-objective or pre-designed quality attributes before you are actually doing or producing. Okay. So here's a schematic or a concept of how quality by design and CMA, CDP, and CQA is coming into play. So this is just for an example that I talked in the earlier slide. So for example, you have the critical material attributes, right? You know that you have a critical process parameter. So you need to analyze or evaluate that how CMA and CPP gonna affect your critical quality attributes. Like for example, the appearance of tablets, content uniformity, right? Strength, dissolution, disintegration time, and so on. Okay. So from CQA, you can specify or set up a quality target product profile that which is a predefined quality profile that you want your product to be, okay, which actually the quality target product profile will affect the safety and efficacy when used in the patients. Okay, so along with that, you can have a quality control strategy and tools, like for example, process analytical technology. And you can also use another tools like design of experiment and the risk analysis to better understand how your CMA and CPP are going to be affect your CQA of the product. So here's just the example of how when you analyze things like the materials input and the process parameter, right? For example, you are going to produce a immediate release tablet. Okay, you list off your API excipient, okay, and the material attributes or their physical chemical properties like particle size distribution, their polymorphism, flowability and compressibility, and so on, right? So after you use a risk analysis or risk assessment to analyze which material attributes are critical, and you can call it as a CMA, critical material attributes, right? So you have a list of CMA. Again, you do the same thing for the process parameter. Let's say in this process, you use a dry granulation to make a dry granules before compress it into a tablets, right? So you analyze what can be your critical process parameter. Let's say, for example, pre-roller and roller compaction, pressure or forces, okay? And milling step, blending and lubrication step, and the main compression force is also a critical process parameter. So right now, you know the CMA and CPP. Okay, so you can use the design of experiment to design, to characterize, and to model how your CMA and CPP are going to affect the quality target product profile by identifying the critical quality attribute. So this is a overall idea of how you better understand your process and your product. So, for example, this is another example of production of a single layer tablet of telmisartan and amlodipine. So the list of the critical material attributes are shown here, right? And they calculate the risk priority number or RPN by probability, sensitivity, and the detectability, right? So the most score, like above 45, 48 score of the RPN will be categorized as a high risk factor or characterized as a critical material attributes. So in this example, you can see that the stabilizer in the formulation or in the materials are critical because they're gonna affect the stability, the solution of the product. So by using the risk assessment, you can know that which material attributes are the critical ones. Okay, so they do the same thing for the disintegrant also. So, and they found that the two material attributes, which are the disintegrant and the stabilizer, has the highest RPN score. So they identify it as a critical material attributes from their formulation. And you do the same thing for the risk analysis of the process parameter. So they list off their process or unit operation, right? Screening, mixing, blending, granulation, drying, and lubrication. And they list off possible process parameters and calculate the RPN score also. Okay, so they found that the kneading time or granulation time is the important one. So this is one of the critical process parameter. And again, another CPP is the speed of the turret or the feeder of the tablet compression and the compression forces. Okay, so 3 CPP should be included into your profile or your. And then they have a list of initial risk assessment results that each critical material attributes, the stabilizer, will affect the impurity in the solution, and the same thing for the disintegrant, which are the one of the CMA, right? And the CPP granulation step or the kneading time or granulation time will have a high effect on impurity and dissolution profile, and also the tablet compression. So here's how when you analyze or identify the CMA and CPP, which going to affect the CQA. Okay. Here is another example. They use a design of experiment to fabricate a 3D printed baclofen mini caplet. Okay, so they use a DOE to decide which materials gonna affect their CQA. Right? So they have a two independent variables: the dimension of the filament making one, the dimension of the equipment, and the infill dimension. They use a 3x3 full factorial design to vary their independent variables and see how things are affected by measuring the dissolution profile time point at 50 and 85 minutes. So they analyze for example, they try to analyze the input variable from the dimension at 5, 7.5, and 10. Okay, and they also have a optimized formulation of their mini caplets also. So this is just an example of using the design of experiment, which is one of the tools of QBD to design your product as you want. Okay, and
I'm sorry, Ajahn, the time is 5 minutes left.
Yep, sure. So you have they also use the response surface to predict the dissolution profile of the product. So here's how things used. So I think I will just give you a brief information about it. So what currently I did talk about is that I in my proof formulate a astaxanthin self microemulsifying delivery system by using the DOE to formulate it, and we can using the DOE to improve its dissolution and absorption in the Charcot cell. So I think I'm running out of time. So if you are interested in more information, you can just taking the opportunity in the Q&A session or just email. Okay.
Okay, thank you for your very nice presentation, Dr. Viakia. Thank you very much. Now we will open the Q&A session. And wait a moment, I will invite Dr. Hideo again. Yeah. Dr. Hideo, are you still here?
Me?
Oh, okay, yeah. Let me pin you first. Wait a moment. Okay. Okay. So for the first Q&A session, I'm asking to the floor, is there any question from the floor? I can see here, oh, Mr. Rido Islami. So please, would you mind to open your camera and microphone?
Yes.
Mr. Rido.
Okay, so can you hear me?
Yeah, we can hear you.
Thank you. So my question is for Dr. Kato. Let me introduce, I'm Rido. I'm also the faculty member of Faculty of Pharmacy at University of Surabaya. So my question is related mostly the animal models that you use as the for the antimicrobial characteristic. So the first question is related to the gender that that you use female instead of male mice. Is it related to the susceptibility of the gender itself to develop the infections? So that is my first questions. And then the next, you want to answer? Can I give one by one or?
I want to one by one. It's okay.
Okay, Dr. Kato. It's okay.
Thank you for your question. So I don't know in detail difference between gender, but historically females is used in animal study. But I showed my presentation in this time. The recent guideline recommend both gender have been strongly encouraged recently, and if established should be considered. So I think you have much money, you try to use both gender in animal study, because I think there may be some difference between genders, but I think it is okay to submit the paper only female.
Okay, thank you, Dr. Kato. For the next question, it's related to the model itself, that you use the thigh and lung, and thigh and lung, right? And then why we need two sites to inject the cyclophosphamide in thigh and lung. Whether is it enough only one site of injection to develop this model? Why we need to do like thigh first and then lung?
Sorry, just a moment, please. Sorry. And the model is two types, thigh infection model and lung infection model. So there are two types of lung and thigh, so different model. So
it's different, or thigh followed by lung?
Yes. And cyclophosphamide in administered from intravenous injection.
Mm-hmm. Okay.
Twice in each model.
Mm-hmm.
Okay.
So if I saw your the table that related to the models of this study, that they did a lot. I think the model that how they develop is they inject the cyclophosphamide to the thigh first, and then followed by lungs.
No.
Is it different model, or just thigh only, or lung only? I just want to confirm that one first.
Oh, sorry. Right now. Just cyclophosphamide is Just a moment. Cyclophosphamide to render the neutral panic. So thigh and lung model is infected site. Lung and thigh is infected site. So just the lung model is pneumonia. Thigh model is skin soft SSI.
For the systemic, right? Maybe
Yeah.
it could be the systemic infections.
Yeah.
Okay. When we maybe I don't know whether you you already did the lung models for this one, because for the lungs, we need to administer the cyclophosphamide with intranasal administration. I still cannot find the information what is the dose of the cyclophosphamide that we have to use for intranasal delivery technique, because that we know that if we administer by nasal administration, it could be possible that the cyclophosphamide can also deliver goes to the brain directly by this delivery system, and then whether the dose is very need to be very low concentration, because it could be toxic for the brain as well. Is it different, totally different for the dose concentration between intraperitoneal and intranasal?
Two different
the dose for intranasal. Is it the same dose with intraperitoneal?
For cyclophosphamide?
Yes. Because
sorry, I don't know in detail, but I think cyclophosphamide has how can I say? I think blood toxicity, such as thrombocytopenia or anemia. So and like hematology toxicity. So I think maybe high dose is administered. The mice have toxicity, then die, easier die, not to infection, okay, not to cause infection.
Okay.
Just cyclophosphamide.
Yeah. Just maybe I have some idea that I don't know, because when I read the table related to the models, they suggested for the intraperitoneal first for several days, and then followed by nasal. That's why they mentioned as the thigh and lung model. At the beginning, I think that it was different model, but I think this is one model that they call it thigh and lung. It's inject with the intraperitoneal followed by intranasal. But I'm concerned as well about the intranasal technique that they use here, because they give the cyclophosphamide, that it could be possible that by this delivery it could be toxic for the brain as well. And then on the other, I think that is my comment so far about this model. It's very interesting, Dr. Kato, because this is the first time I saw that we use two sites of injection to develop the models for the neutropenic in mice. And then the last question, actually, it's just confirmed about T. You mentioned about T more than MIC. I want to confirm what is T, because I don't know T. In your slide, I forgot the slide number, but you've mentioned the parameter T more than MIC. What is T stand for? Why we need to compare T more than MIC?
T T-O, T-O about MRC. T means time.
Okay, type. Oh, type. What is the time for? Oh, I see. I
Time,
get it.
time
Because,
over MRC.
yeah, I'm just thinking that it was like related to the PK parameter, specific
Sorry.
PK. Yeah, it's okay. Just confirm about that one. Thank you so much, Dr. Kato, for your explanation.
Thank you.
Thank you, Dr. Kato, and thank you, Mr. Ridho Islami. So will be the second question will be a fast question for Dr. Pirakyat. I see from Mrs. Angi. As I know, this is a student in Chulalongkorn University. Also, I'm sorry that my speaker is not working well. So what a nice presentation that I learned today. I would like to ask Dr. Pirakyat, is there any limitation of QBD comparing with QBT, quality by testing? And the second one is, does QBD always assure that we can produce every pharmaceutical dosage form well or in a good quality? Thank you very much.
Okay, so regarding limitation of quality by design approach, I think there's very less limitations for the QBD compared to the quality by testing method. But the difficulty of doing QBD is that it is systematic, but it takes like more time and effort from, or knowledge or understanding from the personnel who do the product development to actually understand and like actually doing by hand. So it's not like a limitation, but it's another level of doing the product development. So yeah, the second question, does QBD always assure that we can produce a pharmaceutical dosage from well in a good quality? I think if you understand your CMA and CVP very well, and you explore every input variable that's going to be affect your product very well. So within your design space and your DOE space. So yes, I think yes, you are. It can assure that the your product is going to be of the quality or the requirements.
Okay, thank you. I hope it can answer the question, and we will close the Q&A session with these two questions. If there is any further question, we will accommodate the further question. Would you mind to answer outside the meeting if we still have any question for Dr.
Yeah, sure. So you can just yes, send me a list of questions, and then I'm happy to answer.
Okay. Thank you very much. Thank you very much, Dr. Kato and Dr. Pirakyat, for your inspiring talk. Thank you, and see you again, and good morning. Thank you very much.
Thank you.
Okay, for the third session, I would like to ask is Dr. Muhammad Hafiz Abdul Aziz already with us?
Yes.
Good morning.
Ya,
Dokter.
Sudah. Selamat pagi.
Selamat pagi. I will try to pin you first. Let me find you. Let me try to find you first. Or can the committee help me to pin Dr. Abdul Aziz here? Let me try to find you.
If I've been spotlight, Dr. Hafiz and Mr. Adit.
Oh yeah. You can help me to pin and make it a spotlight, right?
Yes, I did.
Oh, okay. Why I cannot still see Dr. Abdul Aziz here. Okay. Okay. Okay, good morning. Okay, we will continue with the third presentation. Today, I will introduce our third speaker today. So the the third presentation will be with Muhammad Hafiz Abdul Aziz, PhD. So I will read first his education background. His Master of Clinical Pharmacy was done in National University of Malaysia, and then continue with a PhD in medicine in the University of Queensland. Sorry. So for the working experience still present, Dr. Hafiz Abdul Aziz is in the George Institute for Global Health Clinical Research Associate, and so many publication of him also, and the newest publication, as I can see here, the research is about population pharmacokinetics and dosing simulation of ampicillin and sulbactam in hospitalized adult patients. And also we can see here the publication about why is implementation of beta-lactam therapeutic drug monitoring for the critically ill failing short, a multi-center mixed method study, and also several publication of him also. So this morning, Dr. Abdul Aziz will present about the dosing optimization and patients' outcomes. Are you ready, Dr. Abdul Aziz? So, the time is yours. Please, may you can share your presentation.
Okay, so can you see this?
Yes, we can see clearly.
So sebelum mula, so terima kasih, selamat pagi, terima kasih kerana jemputan. Thank you for inviting me. So I'm from UQCCR. So today we are going to discuss about dosing optimization and patient outcomes. All right, so we begin with where do our doses come from? So some important questions for us. Are our doses now appropriate for all patients? Are they optimized for all patients? And what about some patients, like, for example, critically ill patients in the ICU? So as we know, our doses are developed in vitro or in animal models, and then they are scaled for human use. And based on, so they are scaled for human use, and they use healthy volunteers to develop our current dosing regimen. So basically, what dosing we are using now for our sick patients come from healthy volunteers. So the short answer is our current dosing is not optimized for all patients. So this is an interesting systematic review by Goncalves, Pereira, and Povova. So this is a large systematic review by these two investigators. So they reviewed the PK of 6 beta-lactam antibiotics, meropenem, imipenem, piperacillin, cefepime, and ceftazidime. And what they found was really interesting because they reported significant pharmacokinetic heterogeneity in 2 primary PK parameters, which is the volume of distribution and clearance. So when we talk about dosing, we always talk about these 2 parameters. And we could see that, so these are for ICU patients and these are healthy volunteers. So we can see that there are 2-fold variations between the VD and clearance from healthy volunteers and also ICU patients. But the thing is, our current dosing regimen is, so they were mainly developed by the VD and clearance of healthy volunteers patients. So this is also another study, interesting study. So this is from the DALI study, or Defining Antibiotic Level in the ICU study. So they recruited 361 patients from 68 ICUs across Europe. So in these patients, they receive what is termed as conventional beta-lactam antibiotic dosing. So which is the normal dose that we would use in an ICU. And we could see that from that dosing, so we can see that high variability in terms of concentration and also PK/PD target attainment. You could see that if you give a dose for patients in the ICU, you could see that someone would demonstrate a high concentration and someone would demonstrate low concentration. And as you know, for beta-lactam antibiotics, so the PK/PD target for beta-lactam antibiotic is time above MIC. So 50% or 100% of time above MIC. So because we have variability in concentration, that would lead to variability in target attainment. So what's interesting, so you have the conservative PK/PD target, which is 50% of time above MIC, and you also have an aggressive PK/PD target, where you go to target 100% of time above MIC. Even with the conservative PK/PD target, 20% of these patients didn't achieve their target, and these 20% of patients, so they are 32% more likely to demonstrate negative outcomes. So we need a different type of dosing in the ICU. So what can we do? So we can either use unit level interventions where we use prolonged beta-lactam infusion. We could use dosing nomograms like what we are doing now. You could use dosing software, and you could also use therapeutic drug monitoring to optimize antimicrobial treatment. So because I only have 20 minutes, I think I will only focus on prolonged infusion and TDM, which is the two main choices for us to use to optimize antimicrobial therapy for specific patients. So is prolonged infusion or extended infusion or continuous infusion new? So prolonged infusion or extended infusion is defined as any infusion that is extended or prolonged more than 2 hours. So for example, we extend infusion for meropenem for 3 hours throughout the dosing interval, or you could continuously infuse meropenem, sorry, not for meropenem because of stability issue, for piperacillin for 24 hours. So is this method new? So no, because Harry Eagle discussed this in 1953 in his publication. So they tested in a mouse thigh model. So they noticed that as penicillin pre-interval increases, the dose and time to cure the mice also increased. So he proposed to use continuous drips of penicillin or repeated penicillin injection between 2 to 4 hourly injections. So this strategy is not new. But in 1997, the late Johan Mouton probably raised this issue again. So he compared in an in vitro PK model, which mimics human concentrations, IV, intermittent bolus, versus continuous infusion. And what he found was, so after the 4th dose, the colony forming unit for bolus and also continuous infusion, it's higher than bolus. And you would need 4 times above MIC in neutropenic hosts to achieve an effective kill. So this is one of the first biological precedents supporting prolonged infusion or continuous infusion. So that's so we have discussed animal model and we have discussed an in vitro model. So now we look at PK/PD data. So this is a study in 10 critically ill patients with normal renal function. So they were randomized to receive continuous infusion and also bolus. So meropenem, and they measured plasma concentration and also tissue concentration. So we could see that with continuous infusion, the concentration here is stable, and with intermittent bolus, so you would get high concentration and then the concentrations would drop. You see, and this is also reflected in tissue concentration. So and not only in this PKPD study. So all PKPD study shows that continuous infusion or prolonged infusion demonstrates superior plasma concentrations and also tissue concentrations, and this would lead to superior PKPD target attainment. So all PKPD data now shows this. So why don't we change for beta-lactam antibiotics? Because we have in vitro support, we have in animal model support, and almost all PKPD modeling and simulation studies favors continuous infusion compared to the traditional intermittent infusion strategy. Because in outcome studies, in clinical outcome studies, we have mixed findings, but certainly prolonged infusion was not inferior to intermittent bolus. And also in meta-analysis, it's a kind of a mixed bag, but again, prolonged infusion is not inferior compared to bolus. So why are we still reluctant to change our method? So it's because of the clinical outcome studies, the findings were mixed. So it could be due to several limitations. So the patients that were recruited in those studies, so small sample sizes and heterogeneous patient population. So when I mention heterogeneous, meaning we are also including patients, those who are not severely ill in these studies. So prolonged infusion or continuous infusion, we have to remember that they won't be effective for all ICU patients, but only in a subset of patient, which is critically ill patient with sepsis and with infected with pathogens with high MIC, and inclusions of patients with renal impairment. So because beta-lactam is predominantly cleared through renal elimination, so if they are already on RRT, for example, they are not clearing the drugs. So the exposure or concentrations should theoretically be enough to cure the infection. So we would need to target for those who have normal renal function. So severity of disease, so inclusion of patients with low level of illness severity and pathogens with low MIC. So because of this, I hypothesize that prolonged infusion would not benefit all patients, but those patients who come mainly from Southeast Asia or Southeast Asia Pacific region, where we have high patients with high illness severity and also pathogens with higher MIC, like Pseudomonas aeruginosa or Acinetobacter baumannii. So based on our systematic program, we have identified few subpopulations that would benefit from prolonged infusion. So they are likely patients who are not on RRT, patients infected with pathogens with high MIC, especially Acinetobacter baumannii and Pseudomonas aeruginosa, and patients with pneumonia. So from that DALI study, we perform a post-hoc analysis. So in this post-hoc analysis, we evaluated patients on extended infusion, continuous infusion, and also intermittent infusion. And we only included meropenem and PIP test. So based on this study, we found a signal that agrees with our hypothesis, which is prolonged infusion won't be beneficial for all patients, but would be beneficial for patients with high severity of illness. So we could see that here, patients with a SOFA score of more than 9 would benefit with prolonged infusion more with those patients who do not have SOFA score of 9 and above. And also in lung infections, respiratory infections. So we could see some signals. So based on that, so this is a stepwise example of stepwise research program where we build our trail of evidence, right, from bench to bedside, from the lowest evidence, which is in vitro data, animal data, and we proceed with observational data. For example, the post-hoc analysis of the DALI study, human PKPD data and proof of concept trial, BLINK-1, and then phase 2 trial, BLINK-2 and BLISS, and also the ultimate RCT, to investigate whether prolonged infusion is actually better than intermittent infusion in the ICU. So we have the one that is ticked here, we already performed, and we have the data to support a phase 3 trial, which is completed. So just for a brief outline on BLINK-1. So BLINK-1 is just to demonstrate feasibility of to randomize patients to continuous infusion or bolus treatment arm. So it was performed in 60 patients. So we recruited 60 patients across 5 ICUs across Australia and New Zealand. This is how we blinded the treatment. So the patients or the physicians won't know whether they receive continuous infusion or bolus dosing. And this is the results. So basically, no survival benefit was found. But there was cure and PK/PD target attainment separation, you can see here. But in this study, in this BLINK-1, the culture positive infections were really low, and Pseudomonas infections in this cohort was also low, so only 9%. And then we proceed to perform BLINK-2. BLINK-2 is multi-center, double-blind, double-dummy RCT. So only recruited severe sepsis patients receiving beta-lactam antibiotics. The primary outcome is ICU 3 days up to day 28. So we recruited 420 patients across 25 ICU, again, across Australia, New Zealand, and also Hong Kong. But in this study, we also included RRT patients, okay, patients on renal replacement therapy. So again, because we recruited patients on renal replacement therapy, no difference between continuous infusion and bolus dosing in all endpoints. Again, the culture positive infections rate were low, and majority were E. coli and Klebsiella pneumonia, which is likely to have low MIC. So again, this shows that prolonged infusion or continuous infusion won't be beneficial for all patients. And then we proceed to perform BLISS. So BLISS is special in a way that it was performed in a different country. It was performed in Malaysia. So in 2 ICUs, one from KL and one from Kuantan. So the primary outcome is PK/PD target attainment, where we see whether the concentration of piperacillin, meropenem, and cefepime hit the concentration target that we want. And we also evaluated clinical cure after beta-lactam therapy. So again, as we can see, there is no problem at all for prolonged infusion or continuous infusion to demonstrate superior PK/PD target attainment. This is for continuous infusion. This is bolus dosing. So in this study, because we performed this study in Malaysia, we could see some signal. We could see that patients who receive continuous infusion demonstrated higher clinical cure compared to the control or intermittent infusion. And we could also that for patients, we could see the same signals coming. We could see that patients who had lung infection, so prolonged infusion might be better, and also patients who were infected by either Acinetobacter or Pseudomonas aeruginosa. We could see some signals, the same signals coming up. So then we proceed to perform an individual patient data analysis where we combine the data from BLINK-1, BLINK-2, and BLISS. And from this individual patient data meta-analysis, we found that mortality is increased with increasing age, increasing Apache score or severity of illness, the use of RRT in patients with respiratory infection, cardiovascular dysfunction, and the use of intermittent bolus in administration. So as you can see here that in terms of hospital mortality, so it favors continuous infusion. So we could see some signal here again that continuous infusion might not be beneficial for all patients, but only a subset of patients. So these subset of patients. That's why after this, we decided to perform the Blink-3 trial, which recruited 7,000 patients across several countries. Malaysia is one of the country included, and we Australia, New Zealand, and some countries in Europe. And we can wait for the results to see finally whether prolonged infusion is indeed better than intermittent bolus infusion in ICU patients. So that is prolonged infusion. We can also perform TDM in ICU. I'm sure that we have performed TDM for safety, but not so much for efficacy. This is still a relatively new method to improve antimicrobial dosing. So any of the antibiotics, antifungals, and antivirals can be a candidate, but they need to have tick all these characteristics first. So significant intra or inter-individual pharmaco, the drug must show this variability, and you have a defined exposure range associated with pharmacological or clinical responses. You define the sampling time points, and you have assay to measure the antimicrobials. But of course, it will also importantly depend on your resources, whether you have the necessary resources to perform TDM. So this is an interesting study. So this study was performed by investigators from Belgium. So all patients receive 4.5 grams of pip-taz every 6 hours as 3-hour infusion. So they measure concentrations over 7 days. You could see that over the 7 days, the concentrations are really variable. So they are variable between patients and also within patients. So this variability was also influenced by disease severity, Apache score, and also some other factors. So meaning that you can have a patient that receive exactly the same drug, but they will show you different concentration or exposures throughout their treatment period.
I'm sorry, Dr. Abdul Aziz, the time is around 3 minutes left. Yeah.
Yep, sure.
Okay,
So so for TDM, you need to know the PD for the antibiotics for you to optimize the treatment. So if you know this, you also have the magnitudes of exposure that you can target for to optimize the antimicrobials. So what's important here, you can see that these are from preclinical studies, the exposures needed for clinical studies, and these are the exposures needed in clinical studies. You can see that the exposures needed for most of the antimicrobials is higher in clinical compared to preclinical. And so for antimicrobials, we have a lot of data, but for antifungals, as you can see, a lot of boxes needed to be filled. And also for antivirals, we don't have much data to discuss. So I will go to the most current evidence regarding TDM, So this is the results from the target trial. So this is a study which is conducted in Germany, multi-center. So they recruited patients with sepsis or septic shock receiving continuous infusion pip-taz. They evaluated TDM versus no TDM, and the target that they use is 4 times MIC. And what they found was, so target attainment again favors TDM, but no significant difference between TDM and no TDM in clinical outcomes. So you can see here, no significant difference. But you could see that with TDM, so TDM patients reported lower 28-day mortality and higher clinical and microbiological cure, but it's not significant. It was not significant. So this is also another recent RCT in Holland. So multi-center study, 8 ICUs. So they recruited ICU patients receiving beta-lactam antibiotics or ciprofloxacin. What they did is they compared patients receiving model-informed precision dosing with TDM versus standard dosing. So they use the software inside RX to identify what the correct dose should be, and they perform TDM. And then the primary outcome in this RCT is ICU length of stay. So again, no significant difference in clinical outcomes, and no significant difference in target attainment. It's really interesting. So, so the conclusions here is, so this field is still new, especially for TDM when we need more data. And we know that these strategies won't benefit all patients, but they may be beneficial for the most, the sickest of the sickest in our healthcare system. So to conclude, underdosing leads to negative outcomes and potentially antimicrobial resistance. And TDM as a standard of care, ideally, yes, but it depends on our resources and also competing resources. And again, more clinical data, especially in our region. So they are needed to whether to support prolonged infusion or TDM. So I think I'll end my presentation with this slide. Thank you for your attention.
Thank you, Dr. Abdulaziz, for your inspiring talks. So many things that we can get from your presentation. So for the Q&A session, we will start it after the fourth presenter, Dr. Abdulaziz. So please wait a moment. So I will invite our fourth speakers. Okay, good morning, Dr. Usa. How are you?
Good morning. Okay.
Okay, thank you for your coming today. Thank you for waiting since the beginning of our session. So we already have our fourth presenter today. So already with us, Associate Professor Dr. Usa Chailet Kiao. So she has finished her Master of Art in Economics and PhD in Pharmaceutical Economics and Policy in University of Southern California, Los Angeles, California, USA. And now she is working as a Deputy Dean for International Relations and also as an Associate Professor in Faculty of Pharmacy, Mahidol University, Thailand. She also is a Program Director of Mahidol University Health Technology Assessment Graduate Program in Thailand. And her main topics of teaching are pharmacoeconomics and cost effectiveness modeling in the healthcare. Dr. Usa has almost 100 publications also, most of them about pharmacoeconomics, improvement of healthcare system and technology. Her last 3 book publications are about costs associated with adverse drug reaction among HIV or TB patients in Thailand and economic evaluation of screening strategy for latent tuberculosis infection, and also about cost utility analysis of molecular testing for tuberculosis diagnosis in suspected pulmonary tuberculosis in Thailand. So she will present her topic about implementation of health technology assessment and drugs expenditures containment. Please welcome Associate Professor Dr. Usa Chailet Kiao.
Thank you very much, Professor Adi, for a very nice introduction. And I would like to congratulate the organizers for a very successful webinar conference today. And I feel that I am very honored to share my experience in the implementation of the HTA. Okay, let me share my screen. Okay, did you see my screen, right?
Yes, we can see it clearly.
So my topic for today is the implementation of health technology assessment and drug expenditures containment in Thailand. And the outline of my talk, I will include the HTA in Thailand, the milestone, and also the experience in the application of HTA on drug policy decision making. And I will close by the lesson learned and challenges. So first of all, let me give you a little bit background on the health technology. When we talk about the health technology, it's include the application of organized knowledge and skill in the form of devices, medicine, vaccine, procedure, and system, which are developed to solve health problems and improve quality of life. So any health technology that you apply for the patients to increase their survival or improve the quality of life, basically we call the health technologies. So when we talk about the health technology assessment, it's a form of the multidisciplinary policy research that systematically examines the short and long-term, direct and indirect, and or intended and unintended. And when we assess in terms of the medical aspect, social aspect, and also ethical and economic consequences. And nowadays, the economic consequences of the health technology would be very, very interesting because the policymakers wants to know is the health technology cost effective, or how much budget they need to spend for the patients. And when we assess, we can assess since the development of the health technology throughout the process, like diffusion and also the application of a health technology in a technology, or can be a set of the health technologies. So the objective of the health technology assessment is to provide a bridge between the scientific evidences, and also we include the judgment of the health professionals and also the opinions of the patients and general public. And as a researcher, we incorporate all this information and convey to the policymakers to support the evidences for the need of the policymakers. So the HTA serve as the very important tool for policy decision making when they want to know which health technology should be included in the benefit package, or finally they can make the rational health resource allocation. So for the milestones of the HTA in Thailand, it established this kind of knowledge since 1982 because we found the first economic evaluation publication in Thailand. And then during 1982 to 1995, this period is like economic boom. So the government spent a lot of money on the high technology and expensive health technology, but still is limited only in the central area, and still we got the poor distribution and inequity of access. So later, after 1995, we got the economic crisis, or we call Tom Yam Kung crisis. So the economic really bad. So the government tried to cut down all the budget and the cost, and there was the need for the cost containment and efficiency in the healthcare system. So we had the research unit called the International Health Policy Program, which who helped design the universal health coverage policy in Thailand since 2002. So in this case, they try to cut down the budget and provide all the healthcare services to all Thai population under the universal health coverage. Since then, we got the economic recovery, and during this period, we developed the tools for the health technology assessment. For example, we had the organization called Health Intervention and Technology Assessment Program, or HITAP, and which is the HTA agency who responsible for assessing all the health technology for the need of the policymaker. And we also got the database and also the incorporate the HTA information for the revision of the National List of Essential Medicine in 2008. And since 2008, we had the experience in the application of HTA. And in 2017, we developed a postgraduate program in HTA at Mahidol University. So mainly the HTA information are used for the development of the universal health coverage benefit package, and also the development of the national list of essential medicine. So when we asked the policymakers which issue that they consider when they want to adopt the new health technology into the healthcare system, and we can see that from the survey, the policymaker need to know safety information, efficacy, and also quality of life of the patients, and especially they want to know the cost effectiveness of the health technology before they make decision which health technology should be included in the benefit package. And when we ask if they have the economic evaluation, what they want it to be used, and they said the policymaker think that they want to include for the national drug formulary and also the hospital drug formulary. So in terms of the application of the HTA on drug policy decision making in Thailand, the HTA has been used for resource allocation through the development of the national list of essential medicine, and especially for the price negotiation. And when we use the systematic and transparent way of the setting priority for HTA topics, like which drug they want to know the cost effectiveness, and they can send the list of the drugs, and then the researcher can perform the HTA or economic evaluation research to support the policymakers. And we also encourage to use the local data like cost data in Thailand and efficacy data in Thailand. And if data not available, we also can adopt from other countries. So when we talk about the national list of essential medicine, actually it refers to the pharmaceutical reimbursement list for all the health insurance schemes in Thailand. There are 3 health insurance schemes. The first one is like the most privileged health insurance scheme for the government employee and retiree, and also their dependents, which cover around 10% of Thai population. This type of insurance is like a fee for service. It covers almost everything that they spend for healthcare services. And the second scheme is the Social Security scheme, which cover the private sector employee, around 30% of Thai population. And also the third one, which is the most health insurance scheme in our country, is the universal health coverage, which cover 74% of Thai population. And these healthcare services will pay will be paid by the capitation system. But all the 3 health insurance schemes will refer to the same pharmaceutical reimbursement list, which we call the National List of Essential Medicine. And right now we had the current version in 2022, and it covered more than 800 items, which had like a 1,000 dosage forms, and we classify into like 6 categories. And you can notice that the last category, which is like E2 category, is the medicine that are of high cost but are important for particular group of patients, which this type of drug, they need the budget impact and also economic evaluation information to support before it will be included into this category. Mostly very, very high cost drug and need the economic evaluation and BIA information. So when we look at the criteria to decide which drug should be included, you can see that the drug selection criteria is include like efficacy and safety, and also disease severity, affordability of the national health insurance, and also recommended by clinical practice guideline, and also need to have like supporting evidences. And the one that we also need it for the high cost drug, we need the cost effectiveness and the budget impact information. So this is the flow diagram to show that how they incorporate the HTA or the economic evaluation into the decision making. You can see that we have like 27 committee to select the drug into the National List of Essential Medicine. And once they if the drugs are very high priced, they will send the list of drugs to the Health Economics Working Group. And then the Health Economics Working Group will submit the information, cost effectiveness information to the committee. And they will consider all the evidences, and then finally they will announce that drug should be included into the National List of Essential Medicine or not. And if the drug is not cost effective, they will submit to the price negotiation for coordinate with the pharmaceutical industry and try to reduce the price, and so that we will get the cheaper drug. And then if they willing to decrease the price, and the drug will be included into the National List of Essential Medicine. So in the process of the Health Economics Working Group, they have the when they have the list of drugs that they need the cost effectiveness and budget impact information, they will request the public institute like university like Mahidol University or Chulalongkorn University or Silpakorn University, and also non-profit organization to conduct this kind of research about they have the duration around 32 weeks to submit the report to the Health Economics Working Group, and then they will evaluate the quality of the information. And also if they the drug is not cost effective, they will coordinate with the price negotiation working group, and also discuss with the pharmaceutical industry if they willing to decrease the price. And in terms of the HTA, the one that we want to evaluate the economic consequences is the health economic evaluation. And basically it refers to the study that consider both comparative costs and also comparative outcomes, which include the clinical outcomes or health preferences, or we call the quality of life, and also the monetary benefit that we also see the outcome as the money unit, like cost saving. And when we assess, we will compare at least 2 interventions, and then we can finally we want to calculate the ICER, which is calculated by the difference in cost between 2 options divided by the difference in QALY. QALY is the quality adjusted life year. Basically, it's like the number of years that patient can live with a good quality of life, or we call the happy year. And we will calculate the happy year between these 2 drugs. And then finally, if we get the ICER value, we will know that how much money we need to pay more to gain 1 additional unit of the QALY. So in Thailand, if we calculate the ICER and we found that ICER is less than 160,000 baht per QALY gain, it means that intervention would be cost effective, because in the society they're willing to pay for 1 happy year around 160,000 baht. So if the ICER is much more than 160,000 baht, so it means that intervention may be not cost effective. So you can see that this is like an example of the list of drugs that we evaluate the cost effectiveness information. And if you look at the ICER, you can see that if it's cost saving, it means that it can be included into the list. But if it's like the ICER around more than 160,000 baht per QALY gain, you can see that it may not include that into the list. So not only the cost effectiveness information, we also evaluate the budget impact for that intervention, because when the government will need to know how much budget they need to spend for that intervention. So basically, we will calculate the total cost before we include the drug into the list, and then estimate if the drug include into the list, how much budget they need to spend. So basically the total cost will calculate by the number of patients who will have that health intervention multiplied by the unit cost. So you have the total budget, right? And you can compare at the current situation that the drug is not included into the list, and compare to if the drug include into the list, how much budget they need to spend more. So
I'm sorry,
you can see
the time is 2 minutes left.
Yes, I almost finished. And the relationship between the cost effectiveness and the budget impact analysis, you can see that if it's less than the threshold and spend low budget, right, it will be included into the benefit package or the national list of essential medicine. And if even though if it's cost effective, but if they spend high budget, right, they will not include it into the list. So another example that I want to show you is the price negotiation that we got. This is the example that we successfully negotiate the price with the pharmaceutical industry, and we found that only the 3 drugs we can save the budget around more than 1,000 million. You can see that the original price for example, 8,000 reduced to 2,500, then which we can save 152 million. So in summary, lesson learned from Thailand about the HTA, we feel that the role of HTA has continuously played a significant role in drug policy in Thailand, and when we apply the HTA, we need a strong commitment from the chair or from the government, and also the HTA capacity building is very crucial, and also need to apply for the guidelines, for the methodological guideline. And however, there is a need for the formal establishment of the government body and also retaining the capacity of the HTA researchers in the non-profit institutes. So that would be all for my talk. Thank you very much for your kind attention.
Thank you very much, Dr. Usa, for your inspiring talks. So many insights that we can get from your presentation. So we will start the second Q&A session, but I will invite Dr. Abdul Aziz first. I see there is already a question here. Wait a moment. Okay, Dr. Abdul Aziz already with us also. So for the first question, I see Mrs. Sylvie here raising her hand. So Mrs. Sylvie, can you start your question?
Yeah, okay. Thank you very much for the opportunity, Mr. Aditya. My name is Sylvie. I'm from University of Surabaya. Thank you very much for a very nice presentation from both Dr. Hafiz and Dr. Usa. I would like to address my question for both of you. First, Hafiz, I wonder about the results from the study that comparing continuous infusion versus bolus injection. In your study, you mentioned that the results show there is no difference between both interventions. I wonder whether this result apply only in high-income countries, or whether this result also apply in low-middle-income countries, because maybe the burden of infections is different between both settings. And for Dr. Usa, I would like to ask about the method you use to establish the ICER in Thailand. What is the method you use, and also whether the threshold applied for all interventions for all disease or conditions, or for a rare disease, the ICER, the threshold for ICER can be different. Thank you very much.
Thank you, Dr. Sylvie. Dr. Sylvie is also a clinical pharmacist in our university, also in our faculty. So I will give the time for Dr. Abdul Aziz first for the first question to answer.
Yes, thank you, Dr. Sylvie. Exactly. So what we see is those studies which have demonstrated prolonged infusion benefits, usually they come from not the developed world. You see from China, you see from Iran, you see from Malaysia. But unfortunately, most studies currently, evaluating continuous infusion or prolonged infusion versus bolus dosing, they come from high-income countries, European countries, the US. And that's why when we did that study in Malaysia, we started to see some signals that maybe we are looking at different population. So for example, in Australia, the mortality rate in the ICU is 10%. It's really, really low. You can't really reduce the mortality rate anymore. But in some countries, for example, Malaysia, it's 40%. So there are still opportunities. And you are right in a way that the burden of disease is actually high, and the pathogens, they are more resistant to antibiotics, and the MICs are higher. And based on PK/PD data and also in vitro and animal data, so prolonged infusion would actually be beneficial for these patients.
Okay, thank you.
Thank you, Doctor. So please, Dr. Usa, maybe for the second question.
Yes, thank you for your question. Actually, when we conduct the economic evaluation study and finally we got the ICER, you can perform in two approaches. The first one, you can collect the data from the real world practice. But normally and mostly we perform using the economic evaluation or modeling. Basically, you develop the cost effectiveness modeling and then try to estimate and predict what is the expected cost and what is the expected quality, and then calculate as the ICER. And also another question is for the threshold. The threshold we use for the treatment in all diseases, but it is very interesting that for the rare disease, it would be not cost effective, right? Because the very high price and also maybe the patient cannot be cured. So for the rare disease, we don't use the cost effectiveness analysis as the threshold, but we use the budget impact analysis because the rare disease will have like very low number of patients. So when we calculate, the government spend only a few amounts of the budget. So they also incorporate the drug into the list based on the budget impact criteria, not the cost effectiveness.
Thank you very much.
Thank you very much, Dr. Usa. I see the second participant that want to ask also. I can see Mr. Poppy there. Okay, Mr. Poppy, you can ask your question.
Okay, thank you, Mr. Adit, for the opportunity to ask any question during this session. So first of all, I will have one question for Dr. Hafiz. The first one will be, could we use all PK/PD targets for all patients group, including pediatric patients? And then for the second question for Dr. Usa, as you mentioned in your slide that the process to implement the HTA, it takes a long time, including in Thailand. So could you describe the ideal model for collaboration between the government and education institution to enhance the implementation of HTA? Because in Indonesia, we also just implement the universal health coverage in 2014. Thank you.
Okay, thank you, Mr. Poppy. I think this is question for Dr. Usa. I'm sorry, Dr. Abdul Aziz. Okay, can you answer Mr. Poppy question?
Yes, so that is a good question. So basically, most of the clinical data, for example, for most of the antibiotics, so the data, so they come from adult studies. But for example, for beta-lactam antimicrobial, so we still can use that target. For example, for piperacillin, 16 milligrams per liter, but that is for efficacy. So we need to also remember for toxicity. So we need more data to show that we have a range where we can target, which is effectively safe and also efficacious for pediatric patient. But no question, that is also the way forward, and also one of the most important research area that should be explored further, whether using PK/PD data to optimize antimicrobial dosing also applies for pediatric patient.
Okay, thank you. So I have a follow-up question. So
Yeah.
it means that if, for the example, we have a limitation in the PK/PD targets for all patients, is there any factors or other factors that influence PK/PD targets that we need to choose, whether, for the example, for the Indonesian setting and the setting abroad, can we use the same data for the PK/PD targets? Thank you.
So if, for example, let's take beta-lactam as an example. So in animal studies, so for example, you would need around 40 to 70% of time above MIC for antibiotic efficacy. But when you run these studies in humans, you see that for some patients you require higher exposures. So for example, 100 times above MIC. So instead of for piperacillin, for example, so you would require 16 milligram per liters throughout the dosing interval. And this also, as you asked just now, the targets is also different for patients who come from countries or population who have higher illness severity or higher pathogens with higher MIC. So essentially 50% of above MIC is the minimum target if we are talking about beta-lactam antibiotics. But if you come from an area which has severe patients infected with higher MIC, the target should be higher, which means you aim to target 100 times above MIC. But just remember that because people think that right now you need higher targets, so we start to see the incidence for toxicity for beta-lactam, for example, is increasing in the last few years. It's because we begin to using aggressive dosing, but we forgot to think about the toxicity effect.
Okay,
I hope
you very
that
much,
answers your questions. Yes.
Thank you, Dr. Poppy. And thank you, Dr. Abdul Aziz and Dr. Usa, as already answered the question and for your inspiring presentation. So we come to the end of Q&A session. If there is still a question from the participant, we will try to accommodate the next question, maybe in a Google form, some things, and we will answer it outside the webinar today. So, okay, we come to the end of our webinar today. But before closing this event, maybe because we didn't meet Dr. Abdul Aziz this morning, so maybe we will have the second photo session right now. So we have a photo with Dr. Abdul Aziz and Dr. Usa. I hope that Dr. Kato is still here. Dr. Kato, are you still here with us?
Yes.
Okay.
I'm here.
Okay, so we will start the second photo session. So I hope that everyone can open the camera. So please, the committee, can you help us to take a photo of all of the participants?
Okay, Pak Adit. I will help you to take the photos or the screenshots. Thank you for turning on your camera. I will take from the first page first. In 1, 2, 3. And then moving to the second one. 1, 2, 3. Just smile because we don't know where you are, whether it is on the first page, second page, or third page. Okay, 1, 2, 3. I think it is until the third page only, Pak Adit.
Okay. Okay, finish?
Yep.
Okay. Okay, thank you so much for your for our all of our speakers today. Thank you for your willingness to share so many information that we get today. So, and also probably we can learn that we will start working. Our responsibility start from the laboratory until the governmental responsibility. That's our responsibility as a pharmacist. Yeah. Thank you so much once more time for all of the speakers, all of the participants, and all of the committee. There is one information from the committee also that we will get the certificate around 2 weeks after this meeting. So we finally in the end of our event today. Thank you very much for everyone, and happy birthday to University of Surabaya. Thank you once again, and goodbye.
Thank you.
Thank you, Dr. Kato. Thank you, Dr. Usa, and thank you, Dr. Abdul Aziz. Thank you very much.
Thank you.
See you
Thank
again.
you.
See you again later. Thank you. Thank you, everyone.
Thank you very much.
Ada kerja.